everyone. Today we'll be discussing cell division, and that involves mitosis and meiosis. So mitosis is the way by which all of our body cells divide. So that includes all of the somatic cells, except for our reproductive cells. So every cell that you can think of in the body, except the reproductive cells, go through the process of mitosis for the purposes of cell division. Not only that, but asexual organisms like certain kinds of bacteria that don't need a partner to divide, they can reproduce this way. So bacteria just make copies of themselves, which is essentially cloning, and that's the way by which they produce offspring. And again, that's called asexual reproduction. When specific to bacteria, it's called binary fission. This is used for the process of growth and development of organisms that are sexual beings like humans, and it's in order to produce genetically identical daughter cells. So the whole purpose of one of your skin cells dividing is to produce two new skin cells that are genetically identical to the cell you started with. So DNA replication is the way by which the DNA is copied in there, and then it's split into two new cells through mitosis. Meiosis, on the other hand, is used for sexual reproduction in sexual organisms like humans. So this one is specific to our reproductive organs like egg cells and sperm cells. You result in genetically different or distinct daughter cells at the end of the process of meiosis, and it is a much longer process than mitosis. It's basically twice as long. So I'll take you through the two processes together. Starting with mitosis, we first want to discuss the cell cycle. So this is essentially a picture of the different stages that a cell goes through. So throughout most of a cell's life, it is essentially just existing and growing. So G1 and G2 over here are the growth phases, and S phase is essentially when the DNA copies itself. So that's DNA replication, and in this case, it's called DNA synthesis, the fact that you're copying DNA and making new DNA. And that's known as S phase. Now you can see through this gray coloring here, all three of these growth and S phases together are known as interphase. So most of the time, around 90% of the time, the cell is existing in this phase called interphase. And there are three timings within that. Only about 10% of the time, through this light orange coloring you can see here, does the cell actually go through the active division process called mitosis. And mitosis, there are 4 phases to it, and following those phases is the complete separation of the two cells called cytokinesis. So cytokinesis happens immediately following the end of mitosis. Now, when you're drawing mitosis and when you're drawing later on the process of meiosis, you can include drawing interphase and cytokinesis within it. But just be aware that mitosis is actually separate from those phases. In mitosis, you start with one cell and you form 2 daughter cells. And again, they are genetically identical to the one that you started with, called the parent cell. So let's learn some terminology about how chromosomes divide. So firstly, this is a picture of a chromosome, and this chromosome is replicated. So it has copied itself. Anytime you see that characteristic X shape in textbooks or online versions, you can indicate that that is a copied chromosome. So if we go back, that chromosome has already gone through S phase. If it was not copied, it would just be one arm. For example, the arm on the left-hand side. When it copies itself, you generate this arm on the right-hand side, and you keep it together at the center of the chromosome called the centromere. This is where little spindle fibers will attach from either end of the cell to pull this chromosome apart. Because again, it has copied itself. So now you have 2 copies, and you can pull them in separate direction into 2 new cells. These two, in relation to each other, the two arms are called sister chromatids, meaning that one should be a direct copy of the other. So for every chromosome you have, they'll each copy themselves, so they're known as replicated, and then they'll eventually split. When you just have the one arm by itself, it's shown as a single line, and it's an unreplicated chromosome. So it has not gone through S phase yet. Your book has all of the stages in mitosis written out. We are not going to go through pro-metaphase, so you can scratch that one off. Instead, I'd like you to know the acronym PMAT. So PMAT stands for prophase, metaphase, anaphase, and telophase. And those are the 4 distinct stages of mitosis. Some books teach that 5th pro-metaphase step that's in between the first two, but we won't go into that one. So for prophase, it's important to note here is an idealized image of a cell. Here's the nucleus inside. And basically what needs to happen in prophase is that the nuclear envelope has to break down so that the DNA can be released in throughout the cell. These little centrosomes are important for attaching little spindle fibers to the chromosomes to separate them later on. So they start migrating to opposite ends of the cell because essentially the cell will split right down the middle. In metaphase, what happens is that every one of the chromosomes, once they are replicated, they will line up at the center of the cell. So that's known as the metaphase plate. Now those little purple spindle fibers will start to attach to each sister chromatid within each of those chromosomes. In anaphase, each of those sister chromatids from each chromosome will go in opposite directions. Again, it's those spindle fibers that pull them that way. And then in telophase, that's when you start to get the separation of that cell into two, and the nuclear envelopes start to reappear around each of the unreplicated chromosomes. You have a copy here and a copy here. Cytokinesis means splitting of the cytoplasm, so that's when the final splitting occurs in the cell. And if you're an animal cell, that little indent is called a cleavage furrow. But if you're a plant cell, the cells don't fully separate, they sort of remain attached together so that different nutrients and factors can be passed on from one cell to another. So instead, what forms is a cell plate. So if you're drawing the process of mitosis, this might be how you draw it. So if we have a starting cell, our starting cell is always diploid, meaning that it has the full set of chromosomes we're interested in. I've only drawn 4 chromosomes, and the diploid number is represented by 2n. 2n means diploid, equals 4 means how many chromosomes we have. So we have 4 chromosomes here. And so I drew them as little lines to indicate that they have not been replicated yet. Now, if this was a human cell, we would have 46 of those here. Now, for mitosis, the very first phase that you want to draw is actually the one right before. It's called interphase. And all we're interested in drawing is the process of replication that occurs. So chromosomes replicate in interphase, and how you show that is by turning each one of these, each of the 4 chromosomes, into an X shape. That shows that there are 2 arms to each of the chromosome, meaning each one has been replicated. Prophase is essentially the first part or the first phase of mitosis. And in prophase, your 4 chromosomes, apart from other things that are happening in the cell, those 4 chromosomes don't look any different than they would as in interphase. So you draw the same 4 chromosomes in there. Then metaphase happens where each of those chromosomes is lined up at the center of the cell. So we have 1, 2, 3, 4 chromosomes, each of them replicated in the center of the cell. In anaphase, and you don't need to draw the spindle fibers, just what's happening to the chromosomes. In anaphase, remember, this back here, there's 2 arms here. And so each one of those sister chromatids would be pulled in opposite directions. So 4 going towards the left and 4 going towards the right, and they are becoming unreplicated. Telophase is next, and this is where we see the appearance of that cleavage furrow. And again, don't draw things any differently than in the previous picture. We have 4 unreplicated chromosomes on the left, 4 on the right, so that is the same that you draw in the next phase. Cytokinesis is the full splitting of the cytoplasm. So you would draw your 2 cells, and again, the same as in the previous picture, 4 unreplicated chromosomes in each one. And you would label these cells as diploid, full set of chromosomes, 2n equals 4. If you get these 2 results at the end, they should be genetically identical to the parent. So double check. Do you have the same number, and are they the same as what you started with? And they are in this case. So that is the process of drawing mitosis. Again, remember that cytokinesis is going to be a little bit different in animals versus plants, where the plants retain something called a cell plate that keeps the cells very close together, whereas in animal cells, the cells fully separate. Now, if something goes wrong in the process of mitosis, or the cell cycle in general, that can lead to cancer. And because we're really interested in mitosis and retaining genetically identical cells, if you get a mutation and those mutations accumulate, that's how cancerous tumors arise. So there are genes within our bodies called oncogenes that are specifically cancer-causing, and they can be inherited. We also have proto-oncogenes, which are genes that are involved in regulating the cell cycle. So if you go way back to the cell cycle, you can see that there are these division lines between each of the phases. And so each of these division lines actually have really particular genes, which code for proteins. So really particular proteins that come in at that point in time and make sure that we are progressing through the cell cycle at an appropriate rate. If we progress through the cell cycle too quickly, then the cell cycle mutates, and then cells start growing abnormally, as is the case with cancer cells. So those proto-oncogenes are those involved in cell cycle control. There is another set of genes called tumor suppressor genes, and these are fairly well understood as well. They're also to do with cell cycle control, so they'll come in and make sure the cell cycle is progressing appropriately. Now, if it's going too quickly, so if we're going through S phase to G2 without having a checkpoint and making sure we're on the right track, if we bypass that, we grow too quickly, but we also grow abnormally. And so tumor suppressor genes are those that are supposed to stop that cell from continuing to grow abnormally. So one example of a tumor suppressor gene that's really well studied is P53. And P53, through an article in 2016, it was found that using a meta-analysis of every study on cancer that has been reported on, including genetics of cancer, that P53 is a gene that's mutated in half of all cancers, regardless of the cancer. And so what P53 normally does, it codes for a protein, and the protein is called P53, same as the gene name. So this P53 protein is supposed to sense things like DNA damage, so if you get a mutation, cell cycle abnormalities, so if something's progressing too quickly, or hypoxia, not enough oxygen to the cell, then P53 will come in. It'll be activated, and what it will do is it'll stop the cell cycle. So it'll say, hey, abnormal cell, stop. And then it will repair damage if there's damage. Or it will allow the cell to undergo programmed cell death called apoptosis. So essentially, it'll mark it for cell death. That's if it has progressed too far and if the damage cannot be repaired. Now, if the damage can be repaired, and it's fairly simple to repair, P53 will locate the correct repair mechanisms to do that, and then it will restart the cell cycle in hopes that everything is fixed. If you inherit a mutated P53 gene, or if you damage one of your good copies of P53, which can be done epigenetically through environmental factors, as we learned before, then if the cell has damage within it, the mutated P53 protein will not be able to sense it. And so what happens is actually the cell cycle continues, and it continues abnormally. And that's how cells become cancerous. The cell cycle is just going too quickly, and there's nothing there to check it and make sure it's appropriate. Another gene called BRCA1 and BRCA2, those are genes involved in breast cancer. And those are tumor suppressor genes that are supposed to come in and stop um, inappropriate cell cycle control. And in people who have inherited forms of breast cancer, that's the gene that's mutated. It's a mutated BRCA1 or BRCA2 gene that functions in this type of manner as p53. When it's mutated, it can't stop abnormal cells from growing. And so that's what happens with this type of inherited cancer. So moving on to the process of meiosis. Meiosis involves a little bit more in terms of the stages of cell division. And again, meiosis is used for sexual reproduction. So we're thinking egg cells and sperm cells. And something that's important for the process of meiosis is the fact that we have chromosomes that pair together. So you learned already that we have 23 pairs of chromosomes. One comes from mom and one comes from dad. And so what I mean by pairing together is that chromosome number 1, you get one copy of it from your mom and one copy from your dad. So they pair together. So we actually have 23 pairs of chromosomes, and that's important to note for meiosis. So when chromosomes pair together, the two pairs side by side are called homologous chromosomes. Now again, if you take a look at this red X, this is a replicated chromosome again. These would be sister chromatids to each other. If you take a look at the blue, same thing. You would have sister chromatids to each other. But now when you have a homologous chromosome pair, these two X's next to each other, the one red arm as compared to the one blue arm, they are called non-sister chromatids. So they are non-sister chromatids to each other. And that terminology becomes important for meiosis. So here are all the steps of meiosis. Essentially, they're the same steps in mitosis, but they're divided into two parts called meiosis 1, which is then followed by meiosis 2. And so for all of those, prophase, metaphase, anaphase, and telophase, there's a 1, and then there's a second part. So prophase 1, then prophase 2 later on. Cytokinesis does not have a 1 and a 2, but this would be the first round of cytokinesis, and then we have a second round. So your phases are shown in an image from your book that you can have a look at. Everything starts with DNA replication, so that interphase that begins, and then we have prophase 1. Again, we can cut out prometaphase. We have metaphase 1, anaphase 1, telophase 1, and cytokinesis is shown here at the same time. So essentially, DNA is copied in S phase. Then in prophase 1, what happens is that homologous chromosome pairs actually find each other and gravitate towards each other. Like these two, the red and the blue large ones, and then the red and the blue small ones. These are homologous chromosome pairs. And what happens in prophase is actually they come so close together that they exchange pieces of DNA with each other, adding to genetic diversity in the offspring. And so crossing over is what that process is called, and it only ever happens in prophase 1. So it doesn't happen in mitosis, and it doesn't happen in prophase 2. In metaphase 1, homologous chromosome pairs line up together, which is very different than mitosis. In mitosis, every chromosome lined up on its own, but the pairs actually line up in metaphase. In anaphase 1, it's the homologous chromosome pairs that are split apart. And then in telophase 1 and cytokinesis, we still have replicated chromosomes. We haven't unreplicated them, and we have 2 in each cell. So we started with 4 at the beginning of this process, and here we can see that we're already at half the number of chromosomes. So we are haploid instead of diploid. So now we have a second round of meiosis. This entire second round happens exactly like mitosis, but now you have 2 cells. So you have to remember to draw 2 cells. Here's the first cell, here's the second cell. And your chromosomes are genetically distinct because they had that crossover event. So here we have 2 chromosomes in this cell. In prophase, 2 chromosomes in this cell. Then we go to metaphase. They line up individually at the metaphase plate. And then sister chromatids are split in anaphase 2. Now we are unreplicated, and after telophase 2 and cytokinesis, we will have our haploid daughter cells with 2 unreplicated chromosomes in each cell. Remember, each of these cells splits, so we actually get 4 cells at the very end. And the cells are called gametes instead of daughter cells to indicate that they are specialized cells that are either egg or sperm cells. And so if you think about a human, you start with 46 chromosomes at the beginning, but in this process of meiosis, you would end with 23 chromosomes in each of those cells. That means that the egg cell would need to unite with a sperm cell in order to make a full set of 46 chromosomes in the developing embryo. Here's a picture of that crossover event that happens in prophase 1. So you can see if you have a couple different types of genes called A, B, and C, and genes come in dominant versions and recessive versions, dominant by the capital letter, recessive by the lowercase letter, just to indicate that there's variability. If you cross over an arm from one chromosome to another and exchange pieces of DNA, you might suddenly get a recessive version of a gene on one arm that used to have a dominant version, and vice versa. And these are called recombinant offspring or recombinant chromatids, where you have that type of crossover event. So in drawing meiosis, let's say we still start with 2n equals 4 chromosomes. The starting cell is always diploid and always the same. If we go through interphase, the same thing happens. Chromosomes will replicate, so we have 4 X's. And then we get straight to prophase 1. In prophase 1, the pairs start to find each other. You don't have to draw the crossover event, but just remember that it occurs in prophase 1. In metaphase 1, it's the homologous pairs that line up at the plate. And in anaphase 1, it's the homologous pairs that start to split. Telophase 1, we now have half the number of chromosomes going to opposite ends of the cell. And remember, they're still replicated. Cytokinesis ends with 2 cells that are haploid, so we represent that by n equals 2, half the number we started with. But they are still replicated, they're still copied. After cytokinesis, the first round, we dive right into prophase 2. There is no interphase because we're already replicated. So in prophase 2, the cells should not look any different. In metaphase 2, that's where individual chromosomes in each cell will line up at the metaphase plate. In anaphase 2, sister chromatids will start to split for each of the chromosomes in each cell. And in telophase 2, again, we have that pinching off into 2 new cells with 2 chromosomes that are now unreplicated in each side. Eventually, we end up with 4 gametes, each of them having 2 chromosomes, so they're haploid, and those chromosomes are unreplicated. These 4 gametes are now genetically distinct from the initial parent cell, and if you have half the number of chromosomes, then you have done this correctly. So if we take a look at chromosome alignment adds to genetic diversity as well. So there's a few things that actually add to genetic diversity in offspring. So if you think about yourself, you are not a clone of your parents, you are genetically distinct. So crossing over adds genetic diversity, chromosome arrangement adds genetic diversity, and then random fertilization also adds genetic diversity. You already know what crossing over is, but this is chromosome arrangement. So if you think about these chromosomes arranged this way at the metaphase plate in meiosis 1, the blue ones are on the left, the red ones are on the right. And that's how they split, so that these gametes, 2 of them have the blue and 2 have the red. If you look at this picture, let's say randomly if the bottom one switches places. So the red is on the left, the blue is on the right. So now when we start splitting right down the middle, we get a large blue one with a small red one, which is different than this cell, and we get a large red one with a small blue one, which is different than this cell. And then we start splitting in metaphase 2, or in meiosis 2, and our gametes now, based on coloring, look very different than what we have in this chromosome arrangement at the top. And so if you think about how many pairs we have lined up, we have 23 pairs. So the way in which you line up 23 pairs is going to be different, can be different many times. So just that fact in itself, the way we're lining up those chromosomes, that adds a huge amount of genetic variability to our possible gametes. If you want to know that exactly, how we estimate that is through a simple formula. It's 2 to the power of n, where n is the haploid number. So if n is equal to 23 for humans, it's 2 to the power of 23. So we actually, in each of our egg cells or each of our sperm cells, we have for that 2 to the power of 23 possible chromosome combinations, which is around 8 million. So you have 8 million possible different types of cells or gametes. So the genetic variability already from chromosome arrangement is very high. Now, if you take into account random fertilization, that means that randomly any sperm can fertilize any egg. So then you multiply those numbers together. So for an egg cell, it's 8 million. For a sperm cell, it's 8 million. If you multiply those together, you get 64 trillion. So to make hopefully make you excited, you are 1 in 64 trillion possible combinations of chromosomes based on genetic variability. So now if you compare mitosis and meiosis, you can see that mitosis down here is much shorter. It happens similarly to meiosis 2, as you can see from the top part, and it has the names of the same steps like prophase, metaphase, anaphase, and telophase, and both of them start with interphase. We know that meiosis is twice as long, and at the end of meiosis 1, we are already at a haploid cell. And so that those haploid cells continue until the chromosomes become unreplicated. In terms of how many cells we get at the end of one round, we get 2 diploid cells that are genetically identical. In mitosis, in meiosis, we get 4 distinct gametes that are at half the number, so they are haploid. Now, something I didn't mention yet is problems during meiosis. So as you can probably imagine, what can happen in meiosis is here if we look at anaphase 1 or if we look at anaphase 2. So in anaphase 1, what if this little chromosome here on the right-hand side went the opposite direction accidentally? So if one out of all the pairs that were lined up, like for humans, for example, if one of those 23 pairs went the wrong way, what would your resulting haploid cells look like? So that's something to consider as to what chromosome numbers you would get. If you look at anaphase 2, it's the same idea. What if one of those sister chromatids went the opposite direction? So we had one extra going one way and then one less going the other way. If these were the two cases, if we had an event like that, which is called a non-disjunction event, if we had that event happen in anaphase 1, 100% of the gametes would be abnormal. If we had that event happen in anaphase 2, only 50% of the gametes would be abnormal because this top cell here, we would imagine that it would still divide normally. So that's if we had one non-disjunction event. And those non-disjunction events lead to disorders such as Down syndrome, where we have an extra copy in a cell of chromosome 21. And the cell will tolerate an extra copy of a chromosome. Most cells will not tolerate one less chromosome, except in the case of your sex chromosomes. So there are females that have only one X chromosome, and they do have a disorder called Turner syndrome, which includes poor secondary sex characteristics and sterility. There are some females who actually have 3X chromosomes, so they have one extra, kind of like with Down syndrome. And there are no known effects in the females. So as of yet, none have been noted. Likewise, there are males who have an X and a Y and an extra Y. And there are some males who have 2X's and 1Y. And so in the case where you have 2X's and a Y, that's called Klinefelter syndrome. And again, the male has poor secondary sex characteristics and is sterile. The XYY male, there have been no noted effects. The only thing in the literature is increased aggressiveness. But as of any disorders or diseases, none have been noted. There are a few other instances where cells will tolerate extra chromosomes, but generally, for most of our autosomes, the cells will spontaneously abort, such that, or if incorrect chromosome matching happens between egg and sperm, the embryo will be miscarried. So those instances do occur as well. So again, this is another picture comparing mitosis and meiosis. Important things to keep in mind is that crossing over only happens in meiosis, specifically prophase 1. Homologous chromosome pairs line up in metaphase 1, whereas sister chromatids line up in metaphase 2. And homologous chromosomes, that's not important for mitosis. They don't line up. They all just line up individually, which is where you get separation of sister chromatids. And again, the outcome is really important between the two as well. So this is the life cycle of a diploid organism in terms of cell division. We are diploid organisms represented by the letter number 2N once we form as an early embryo called a zygote. We have a total of 46 chromosomes, and they keep dividing to keep our bodies maintained. In our ovaries and in our testes, depending on if we're female or male, that's where meiosis happens. And so all of the eggs are haploid, all of the sperm cells are haploid. When they join together in fertilization, that's when we get a diploid organism or a zygote developing into a full individual. And at that point, mitosis is how the body cells divide. So if you have any questions on cell division, please let me know.